Hello, everyone, and welcome to PAPERI Webinar Series. And my name is Mr. Hagai Kipuyo, a medical scientist and a bioinformatics scientist from Tanzania. And I'll be your moderator for today's session. Thank you for joining us for this session today. And we are delighted to have with us Dr. Eva Aluvaala. Dr. Eva Aluvaala is a senior research scientist at Kenyan Medical Research Institute, where she's the head of Human Identification Laboratory at KEMRI and a leading facility for DNA testing services in Kenya. She has a bachelor's degree in biochemistry, master's in molecular medicine, and PhD in biochemistry, majoring in human population genetics. Her research is focused on the development of human mitogenome reference database for the Kenyan population with application in human health, anthropology, and forensics. Her work is lying in the foundation for key national genomics resources expected to support Kenya's National Genome Initiative while contributing advances in population genomics, precision medicine, and forensics DNA application in Africa. Additionally, Eva is a founder, member of Human Genetics and Genomics Society of Kenya, a non-profit organization that seeks to create awareness and building capacity in human genomics and research in Kenya. She currently serves as the chair. So in today's session, Dr. Eva Aluvaala will talk about scaling human genomics in Kenya, building capacity, data, and health impact. So please join with me in welcoming Dr. Eva Aluvaala. Meanwhile, you can take note of every question that you have, and later we're going to have a Q&A session where you can just submit your question, or you can use the hand function, where you just ask your question. So we can start right ahead. Dr. Eva Aluvaala, please, the floor is yours. You're welcome.
Thank you. Thank you so much, Hagai. Thank you for this opportunity to make this presentation. And I'll just go straight and share my presentation. You can see my presentation?
Yes, we can.
Okay, so I can proceed on?
Yes.
Okay. So thank you once again to the organizers of PAPERI Webinar Series for giving me this opportunity to make this presentation today. As introduced, my name is Dr. Eva Aluvaala Nambati, and I'm a senior research scientist at the Kenya Medical Research Institute. And today I'm going to make a presentation on scaling human genomics in Kenya, building capacity, data, and health impact. So I'd like to start this presentation by trying to really define what scaling human genomics entails. So I'm sure for most of us, when someone would ask you what scaling human genomics is all about, the first thing that would come to your mind is, sequencing of more genomes. But okay, this is partly true, but this is not the entire picture. There's more to it than just sequencing more genomes. And what scaling really entails is being able to move from, those small isolated projects to now have a national genomic initiative. It would also involve building national genomic data sets and making sure that these data sets that you are generating actually represent the genetic diversity that is present in that particular population that you are studying. So scaling goes beyond generation of data, and it should also include expanding community engagement so that the people who are being studied are also aware of the value of genomic research and how it could also benefit them. And the discussions that you have around genomics should not just be limited to the scientists who are conducting the research, but it should also include a broader audience. And where it is possible, these conversations should even be or converted to a national dialogue so that everyone is included and has a voice in the discussions. It would also involve growing the genomic medicine workforce. You need to have more medical scientists, clinical geneticists, bioinformaticians, as well as genetic counselors who are really rare in our ecosystem. And further, it would also involve ensuring that there's equitable access to genomic medicine to everyone. This means that you move from very few people benefiting from it to population-wide impact. That is access, for example, of genetic testing and precision medicine should not just be for a few, but for all that would require it. So scaling is not just about sequencing more DNA, but it's more about translating genomics so that there is population level impact. So what is the genomic opportunity in Kenya? So as you might know, Kenya is recognized as the cradle of humankind. And over the years, it has contributed to studies in human evolution as well as migration studies. And it is home to the three major ethno-linguistic groups that we have in Africa. That is the Bantu, the Nilotes, and the Kushites. So all these groups that you would find in other countries in Africa are present in and represented in Kenya. And in addition, these populations are in diverse environments. In Kenya, we will find the coastal regions, the arid desert-like regions, and even tropical rainforests. So the environments and that we have in Kenya are actually quite diverse. And these have been driving unique genetic adaptations over the years. And because of this, Kenya is actually considered to be one of the most genetically diverse populations in Africa, and it is therefore potentially a rich resource for us to study human genetic diversity or variation. But despite this knowledge, there is still very limited genomic data that has come from the Kenyan population. So there's really a big opportunity here. If you're able to advance genomic medicine in Kenya, it means that we'll be able to get critical insights that will enable us to see the drivers of genetic disease or conditions that probably are not very available right now. And such insights will be able to help us benefit globally and not just in Kenya. So why is it important for us in Kenya to actually scale human genomics? First of all, in the previous years, the disease burden was mostly on infectious diseases, but now in the previous years, we've seen a rise in the burden of non-communicable diseases, and this includes cancer, diabetes, and cardiovascular conditions. And these diseases, they all require a genomic approach for us to be able to now understand the risk in the population or the person and the prevention strategies, as well as how we can now apply targeted treatments. There's also been an increase in the need for rare disease diagnosis. And because many of these diseases have a genetic basis, would need to use genomics, and scaling would then enable us to accurately diagnose in a timely manner, and such diagnosis will enable the clinicians to better support the patients. Apart from this, there's also a growing demand for precision oncology in Kenya. People are now well aware of the impact that precision medicine now has in cancer treatment. But for us to be able to benefit from this, we would need to have our own local genomic data so that we can fully benefit from the advances in precision medicine and actually be able to get personalized care for cancer patients in Kenya. So currently, I would say genomic medicine is still emerging in Kenya. It's still a new concept or an idea, but there is a very fast expanding and growing research and diagnostic infrastructure. People have invested in next generation sequencing equipment. There's growth in bioinformatics capacity, and this cut across both in the public and the private sector. So in terms of infrastructure, I would say Kenya is really well equipped. There's also increasing awareness and uptake. You'll find that now in Kenya, people are more aware. The clinicians are now requesting for these tests, and even the patients are inquiring and asking to be to have these tests done on them to answer their disease or their diseases. Yeah, but currently there is still a gap because we are yet to integrate the use of genomic medicine in routine healthcare. There's no policy or a government policy document to guide on the use of genetic medicine or precision medicine in routine healthcare. And these tests are pretty costly and accessibility is limited. I would say there's only maybe a few high-end private hospitals that would probably offer these genetic tests, but then most of them would still outsource these tests and have them done in abroad or outside countries. Further, we still have, in terms of research, our research is fragmented, and our databases or our data sets are not consolidated in one place. So in order for us to begin addressing the issue of genomic gaps in Kenya, in 2021, we initiated a pilot genomic project in KEMRI, and currently it is the most comprehensive genomic resource that we have in Kenya. And through this pilot project, we were able to establish a biobank that has approximately 900 samples, and these samples were derived from 42 ethnic groups in Kenya, and it covered all the major ethno-linguistic groups. That's the Bantus, the Nilotes, and the Cushitic groups. And from these samples, we have so far managed to generate a mitogenome reference database. And it is the most comprehensive in Kenya to date. And this database has applications, currently applications in forensic genetics for people who are interested in anthropology and population history, as well as medical genetics. So this pilot genomic project is actually laying a foundation for us now to be able to scale and have a national genomic reference database that will not only be for the mitochondrial DNA, but for the entire genome, human genome. So these are some of the insights that we got from the pilot genomic project. From the data that we got from the project, we were able to see or to reveal rich maternal ancestries. And as expected, most of these lineages were of African origin, from L0 to L6, but we also had minor non-African haplogroups in this data set, and this included the H, M, N, R, T, U, I, and K, which is pretty diverse, and this is can be attributed to historical admixtures in the population. But from the African lineages that we had, we also found that it's only 4 that were most prominent, that is L3, L0, L2, and L4. And this made up to 83.9% of all the lineages that we observed in the population. But L3 was the most prevalent at 39.7%, almost 40% of the Kenyan population. We found that most of them were of this lineage. Although now we would find that on these lineages, there's a lot of subgroups and diversity as well. So in summary, we found that there was high genetic diversity in the Kenyan population, but we also saw and observed that maternal ancestries, we found people from different ethnic groups in Kenya would have the same maternal ancestries. And this showed that maternal ancestry is not always equal to your ethnicity. What you define yourself based on your ethnicity is not necessarily what would be your maternal or genetic ancestry. So genetic ancestry actually transcends, or rather goes beyond ethnicity. Apart from this, we also observed that there was clustering of some of these haplogroups in particular a geographical region. For example, you'd find a particular geographical region has more of L4s than L0 and vice versa. So apart from just characterizing the haplogroups that we observed in the population, we also looked at the pathogenic variants that were in the mitochondria. And we found some that were linked to rare diseases, cancer, as well as mitochondrial disorders. Out of these variants that we observed in the mitogenomes, 92.3% were variants that were considered to be non-synonymous, and only 7.7% were synonymous. From this, we would see that most of the variants that we're observing are likely to be functional or to have a pathogenic effect. But interestingly, out of these non-synonymous variants that we observed, only 24.6% were actually present in the MitoMap database, which is the reference database for mitochondrial genomes or variations. So from this study, we were able to demonstrate that most of the variants that could potentially be pathogenic from the Kenyan population are not present in the global database MitoMap. And this means that there are pathogenic variants that are in the Kenyan population that could possibly be causing a disease, but it is yet to be classified. So this can limit the accuracy of diagnosis as well as interpretation. So from this study, we can clearly see that there is a strong case for us to now scale human genomics in Kenya, and not just focused on the mitochondrial genome, as well as but you should also be considering the nuclear genome, because for sure, when we sequence or type the nuclear genome, we are going to discover or see more variations that are significant and are yet to be discovered or classified. So what are some of the challenges that we encountered when we were running this pilot project? For one, we had challenges in recruiting participants, particularly in the remote areas, because of access. It's not everywhere in Kenya that maybe you have good roads. Sometimes there's language barrier. Okay, our common language is Swahili, but if you go really in remote areas, you find that people who don't speak any of the national languages and they're just speaking their ethnic languages. So we had to get like people who could interpret for us so that we could get the samples. But sometimes you're not really sure whether that person has really understood what informed consent is, or they're relying on that interpreter to sort of convince them or tell them that it is okay to participate in this study. We also had limited funding for this project. And from experience, even in my institute, we didn't really have clear guidelines on how we should share this genomic data. We generated the data, but when it came to sharing, we could clearly see that there's limitation and people were still hesitant. They're worried about, okay, this is mitochondrial genome data. It's not just you, it is everyone in your ancestry. If we share it out there, how sure or how safe are we? So up to date, we are still limited, and we are still looking forward to have clear guidelines on how we are going to share genomic data, even as we plan to scale. We also had challenges with data storage. Okay, this is a small project, but still within our institution, we didn't have like a centralized place where we could now deposit this genomic data. But from this pilot project, we were able to sort of set up a repository, and we hope that this will now continue to grow. So as Kenyans, are we really ready to scale? So for us to be able to assess whether we are ready as a country to scale in human genomics, the Human Genetics and Genomics Society of Kenya recently convened a symposium, our second symposium. And during this symposium, we conducted a stakeholder survey, and this was held in February 2026, just this year. And from the stakeholders, from the survey, we were able to see that 57%, which is the majority of the respondents, they felt that Kenya is not really ready to scale, while 43% thought that, okay, we are sort of prepared. And this showed that there was mixed feelings about whether or not we are ready as a country to scale. And it was also clear from the symposium that Kenya had made notable progress in terms of building capacity in research, but it still remained fragmented in terms of the data that we are generating. And we had limited policies as well as weak coordination across systems and the institutions. So from the symposium, these are some of the gaps or issues that were raised. The stakeholder or the respondents felt that before we could move on and scale, there was still a need for us to have a stronger genetic data privacy and protection framework. We still needed to have a well-defined informed consent framework, and they felt that we needed to have standardized legal agreements, the material transfer agreements and data transfer agreements. And also there was concern about data sovereignty and how we are going to safeguard this. Apart from this, people also raised concerns about genetic discrimination, as well as the need for us to have clearer governance on biobanking, and for us to also enhance our ethic review capacity, because it was felt that as it is constituted right now, it would probably not really, they don't really have the capacity to review human genetics studies, and they need to be enhanced in this area. So it was generally felt that if these issues are addressed, then Kenya would be ready to now scale human genomics. From the meeting as well, it was felt that the most urgent thing for us to do was to now develop a national genomics policy, possibly in the next 1 to 3 years. And this needed to not just be a one institution affair. We need to have a multi-sectoral approach so that the Ministry of Health in Kenya is involved, our Data Protection Office, NACOSTI, which is the research regulatory body in Kenya, as well as the relevant research institution. Basically, they all need to come together for us to now develop a national genomics policy that will enable us to now be ready to scale in spaces that people feel are safe and have guidelines. From this discussion, it was felt that the national genomics policy should include issues such as data ownership and stewardship, biobanking, and long-term use of the samples that are in the biobank. And also, it was discussed and agreed that we would need to have policies so that the use of precision medicine is incorporated into clinical care. As it is right now, we don't have policy guidelines that would enable clinicians to now use genomic medicine or precision medicine approaches in clinical care. So again, it was felt that if you have these measures in place or this national genomics policy, then Kenya would be in a position to scale. And finally, I'd like to end with this parting shot to just remind you of what I've talked about in this presentation. So in Kenya, yes, we do have high genetic diversity. We have early data. We have growing capacity and interest in precision medicine and genomic medicine, but we still have gaps, particularly in governance, coordination, and there's limited access to genomic medicine. And these are the barriers that we have that are limiting us from now scaling. And that scaling should not just be a scientific ambition, it needs to be a national initiative. And if we do it right, then it has the potential to transform healthcare in Kenya. So looking forward, we need to build trust in our core communities, strengthen governance, and develop national frameworks that will ensure equity and health impact. So the question for us is no longer whether we need to scale human genetics in Kenya, but rather how quickly and how responsibly are we going to do it. So that is the end of my presentation. And I would like to thank or acknowledge KEMRI for providing the facilities that enabled the pilot project to take place. at the government of Kenya for funding this initial pilot project, and the awesome staff at HID Lab. I would especially like to thank Miss Eunice Gidua, who is an MSc student, who really worked to do the pathogenic variant analysis, and of course the organizers of PAPERI Webinar Series for giving me this opportunity to make the presentation. And with that, I say thank you. I'm open to questions.
Yeah. Yes, thank you so much, Dr. Eva. Now, we'll just receive any questions and comments from anyone. So we will open the floor. If you have any question and any comments, just please use the raise hand feature or type your question into the chat, and I will address as many questions as possible within our allotted time. Yes, the floor is yours. Anybody
I can
with
stop
a question?
sharing, or let me just.
Yeah. Yes, as we are waiting for questions from the audience, maybe I would like to ask you a few. We've heard of what you've talked about, scaling, population level impact, the pilot project itself from KEMRI, the groundbreaking information you've obtained from the SNPs, the structural variants, the assessments, and the gaps. I was more particularly interested in the ethical side of it because, you know, genomics is quite a difficult subject to address in terms of ethics. And maybe we'll just ask, like, in the future prospects, what would you be thinking? What would KEMRI be thinking in terms of you said something about funding, and most funders would require that your data be publicly available, and here you're talking about mitochondrial DNA data, which is it's quite a sensitive issue. So what would you be thinking in terms of that, like funding and the rules that come with it? Because most of countries now would limit themselves into progressing to human genomics due to the high risks, especially in the ethical side of it. Yeah.
I think that is what I was saying. Even from the symposium that we had from the Human Genetics Society, it was felt that we need to address the ethics component. So we need to have that national framework, genomics framework, or policy that would not then guide us. As it is right now, we don't really have that. So people are still a bit hesitant. They are unsure. And sometimes even if when you go to the counties or to the villages, yeah, they're still suspicious. They're not sure. You're just an independent scientist who is coming to them. How sure are they that they are safe? But I think if it becomes a national thing, then everyone is aware and people will be more and more comfortable about it. Just knowing that it is not a personal project, but it is a country or a national project that they need to participate in and they need to feel safe about it. Yeah. Have I answered?
Yes. Yeah. And there's a question here on the chat, and this is from Jihan. I think I'm pronouncing your name right. Forgive me if it's wrong. But he says, thank you for this presentation. Since there are no clear guidelines, on data sharing in your country. What interim ethical framework did you use? And also, of centralized repository, how was the data protected against loss or unauthorized access during your pilot phase? Yeah. Maybe you can attempt to that as we wait for some more. Did you get it, Kelly?
Yes. As I indicated in my presentation, we had challenges with data sharing. And as it is right now, the data is just in KEMRI. We've not shared it to public databases yet. We tried to work on data sharing agreements between the institutions, but we sort of hit a snag. But we are working on it. We hope that we'll eventually be able to now share some of that data, because we also had collaborators that will be able to share that data, and finally we'll be able to have this data in those public repositories or databases. But in the meantime, we have our own repository within KEMRI where the data is stored. People can actually request for this data. You cannot download it, but you can request for it. That's in the interim.
Oh, thank you. I was about to ask if one is interested to make a research on the data you have. Yeah, but that answers my question. And let me just remind the floor that we are into Q&A session. So you can just use your raise hand function, or yeah, just write it on the chat. But yeah, I see there is something on the chat. Let me look at it quickly. Yes. Keith says, thank you, Dr. Eva. Nice presentation, amazing data in your presentation. You mentioned that the genetic diversity has not been captured internationally, and that might affect treatment, especially for cancers. What is your opinion on this?
At this point, I think I'm like speculating. If we found like 90% of the variants in the mitogenomes that we sequenced are potentially pathogenic, but out of these, only was it 23% are in mitomap, meaning that about 60% are yet to be included in mitomap, which is the reference or global database for mitochondrial DNA. So it means these 60% of the variants in our population are yet to be classified or related to a disease. So we don't know exactly which disease it should potentially. We don't have a reference. These variants are not yet in mitomap. So if someone was trying to diagnose patient from the Kenyan population and they don't get the 24% that is already on mitomap, how would you explain the variant that you have observed in that person? You'll not be able to, because that variant is not yet included in that database. So that is the challenge that you have. We could be having disease variants, but they're not yet in a database. So it's not useful. It's not fully useful for our population. It would only apply as it is right now to 24% of the variants that we are observing. Probably not all of them are pathogenic, but I'm sure some of them are, but we've not yet classified and they've not been included in the reference database. Over.
Yes, thank you so much. I would just say maybe like on what you said, there's a lot of scientific questions that could bring up groundbreaking discoveries. So if someone is interested, maybe could they reach you on the same note, like on the same data you have? There are a lot of things that like speculations on the analysis and what maybe they could reach you and come up with something that potentially like partnership into analyzing, and yeah, are you open to that?
Yes, they can reach out to me.
Oh, thank you.
And we can discuss what they would like to do.
All right.
And also if they want to do their own mitogenome DNA analysis for their population, I would be happy to share my protocols.
Exactly. Thank you.
Yeah.
So there's another question, and this is from Dr. Wafa. You mentioned limited policies for data sharing. What would an effective and context-appropriate data sharing framework look like for Kenya? Yeah. And the other one from same.
There's another one. Okay.
Yes, there's another. Should I let you respond to the first?
Just yeah, sure. So for data sharing frameworks, out of my experience, the issues that were there were, for example, if we share this data out there at this point, how sure are we that this data will not be used for other purposes out there? Yeah, we are not sure. So if we had proper guidelines and even means of maybe doing follow-ups, because sometimes you say that you share data and you can only use for this particular research project, but you find that people still go further and dig in and probably use it for other purposes. So we were like, okay, if we share this and it's mitochondrial DNA and it has even forensic applications, how sure people are like, how sure are we that it cannot be used also against us? So are we comfortable? I've given my mitochondrial DNA, but it's for my entire maternal lineage. It's not just me. Are we exposing ourselves or not? So it's partly awareness or understanding of what this is going to be used for, or people are a bit suspicious still. So the other issue was I've talked about sorry, I think I lost that point. I should have written it down. So maybe give me the other question as I think about it. Yeah.
Okay, no problem. So the other question is, given these challenges, what are some realistic steps that can be taken in short term to move forward? And also, are there examples or models within Africa or globally that Kenya could adapt to strengthen its genomic ecosystem?
We are talking about just the first part, the challenges that we encountered.
Yes, what are the realistic steps that could be taken in short term to move forward?
I think there's no shortcut. I think there's no shortcut. We just need to develop that national framework, genomic policy framework. And with that, we'll have the guidelines that is necessary for us now to be able to scale. Because if we don't get this right, then it won't work. I don't think it will go. We'll be able to scale as we should. Yeah. So there's no shortcut. We just need to develop those policies and guidelines.
All right, that's a good response. And to the second part, where are there examples or models that Kenya could adapt to strengthen its genomic ecosystem?
I'll pose that question back to the audience as well. Are there any in Egypt? Because as it is right now, I am not really aware of any, particularly in human genomics. I think there are initiatives in different countries, but I know there are efforts to now consolidate so that we have like initiatives that are talking to each other rather than just having national initiatives that are like silos in those countries, so that even within Africa we can be able to share our data.
Okay, thank you. Maybe for me, I would just like to pose a challenge, and this is really for everyone. Like, we're so limited and scared to move into the human genomics. But what are the trade-offs? What are the benefits? We should weigh them, and maybe we should look into it carefully, because the most concerns will be the ethical parts and the data sharing. But if we really want to, like, for example, into the non-communicable diseases, like if we want to curb the effects of these diseases, we should really look into human genomics. And that requires a lot of genomic data and sequencing and what. So again, this does not neglect the fact that we have to look into these ethical policies and frameworks, and maybe sometimes we would need to adapt with caution and modify to our own context what others have done outside. Because for here, you are talking about Kenya, but it's more of like Africa. You are representing Africa, because most African countries are similar to, like human genomics. Talking about human genomics is it's like a new concept in Africa. So yeah, that would be my challenge to pose, I'd say. Yeah, so to the next question by Sharon, how close are we to have clinically usable reference data, and what's the timeline for its impact in diagnostic interpretation for Kenyan patients? Oh, okay.
Yeah, I would say we are just starting off. As far as I know, the data set that I have is the only reference data in Kenya. But if you're able to scale, the sooner we then now have reference data for our population. Yeah. And Sharon, you're part of our society, so we should be working towards this together. Yeah.
Yeah,
Yeah.
exactly. Thank you, Sharon, for such a question. And we have one again from Jihan. And if you could implement a single structural intervention to optimize operational efficiencies of current genomic workflow, which bottleneck would you prioritize?
I'm not sure I understand that question, or maybe I'm not in a good place to answer it. Just repeat it.
Okay, if you could implement a single structural intervention to optimize the operational efficiency of current genomic workflow, which bottleneck would you prioritize? Maybe if you need some more explanation, Jihan, maybe you could, if you're in a good position, you could explain what you meant by that.
Yeah, please. I think I need further explanation. I don't think I really got it.
Yeah, so Jihan, if you're in a good position, kindly unmute your mic and explain what you meant by that. Or you could just type it on the chat again to explain what you meant.
Hello, Eva. Hello, everyone. So my question is like, actually you have a lot of gaps, a lot of difficulties, etc. So if you could implement just one single intervention to try to optimize and to make it good and a good operational efficiency for your current pilot phase, your current genomic workflow, which which factor would you prioritize? Which part of this genomic workflow would you prioritize? Like trying to prioritize something about technical part, for example, for infrastructure, or trying to talk, for example, with some policymaker in order to make some local and national guidelines, or in which part would you start doing doing something better?
I would say the centralized database, being able to now house our own data in KEMRI, and for people to now access it from there, so that we feel that we have ownership of our data, and we can share from there. Because as it is right now, we didn't really have a centralized place for storing this data, and we anticipate it's going to grow. And if we scale it now to the whole human genome, it's going to be worse because it's bigger. So at this stage, I would prioritize on this to ensure that we have an infrastructure that would be able to now house the data that we are generating, so that also in future we can reuse it, we can use it for future research. Yeah. So that we don't have our data sets for the Kenyan population scattered all over. Yeah.
Thank you. Nice reflection. Thank you very much.
Thank you.
Yes, do we have any more questions? And
hello.
As we are waiting. Yes, hello. You're welcome.
I have a question, but I just need like 2 minutes to be able to structure it, and then I'll ask.
No problem, Lucy. We will be here, but on the allotted time. We are waiting for you. Be happy to receive your questions. Thank you. Yeah, so you can just raise your hand on the raise hand function or type it on the chat. Yes, maybe I would still have a question on the analysis. Like, what reference did you use? What did you use as a reference to analyze the
for the mitogenome? For the mitogenome, it's the revised Cambridge reference sequence. Yeah. That's what we used.
Yeah, that's good, because as of my little knowledge, I go through a lot of human genomic studies, and one of the most challenging issues is that we don't have African reference panel. Okay, we have what is being created by some of the African group, and it's really challenging to infer African, like we are inferring from the basis of European data.
Exactly.
And so sometimes, yes, and this would really be a good step of what KEMRI has done, because being the first is always challenging. But once you're there, you keep on improving from whatever you have. So like it's a very good step that we are actually making references from our own data. But speaking about precision medicine, it's very hard to have a precise like precise information from an African ancestry genome inferred from from the European data. Yeah. So most of the studies that we still sequence, we make analysis, but how possibly would our analysis, like I would say, maybe we would, if we get an African panel reference, maybe we would find variations in all papers that have been published. I don't know, I'm just speculating. I don't know, what would be your comment on that?
Yeah, that is true. And it would be interesting for us to now have the African panel, but I think we'd have to work together and come together for us to be able to do that. And I'm excited that these initiatives are now coming up through the Africa Society for Human Genetics. Yeah.
Yes.
Otherwise, it is as though is it shooting in the dark? You can't really
Exactly.
pinpoint. You don't have evidence of sorts. You are just thinking that it could be, but
yes.
you're not very sure.
You're not very sure. Yeah.
Yes. Yeah.
Yes. So you're welcome, Lucy. I see your hand raised.
Yeah, thank you. Good afternoon, everyone.
Good afternoon.
Good afternoon.
My question is to Dr. Eva. Do you have a lab where you're able to do a metagenomic study on soil microbiome? And what is the turnaround time for that service? And what is the cost for that service?
In KEMRI, we have several labs with the capacity to do metagenomic studies. So maybe you can side chat after this. I'm not sure. Most of them are not service oriented. They are mostly for research purposes. So I can't put a figure to how much they would probably charge you. But what I know and what I've seen, most of them would work as collaborators rather than charging you a fee to run your samples. Yeah.
Yeah,
But
that's
the capacity is there. Yeah. Mm-hmm.
Oh, that's very interesting because I'm doing a study where I need to do metagenomics for the soil microbiome, and we are having a challenge on where we can be able to access the lab that can run for us that analysis. I think it's a good thing to know that we can get the service at KEMRI.
Yeah, the limitation would probably be if that lab at this point is not doing this particular analysis, you would have to maybe buy your reagents, and then they can now run them for you. Because the lab is not the labs are not service based. They don't stock up on reagents to now commercially give a service. But the equipment, the staff, the capabilities are there. So you need to factor in a budget to actually buy your reagents and consumables. And I think that is what has been driving people to send samples abroad. Because I don't know, how many samples do you want to analyze, for example?
Just one. I think the site is one. The initial study included 3 sites, but for the moment, the site will be one.
Yeah, okay. So you see, if the samples are not many, the lab, if they're not routinely doing that test, they would probably have to charge you a lot for them to cover their costs. Yeah, but we can side chat. We inquire and see if it works for you.
I'll do that. Thank you.
Yeah. Can't hear you.
Hagai, please
Hagai.
unmute yourself. Unmute yourself, please.
Sorry. Thank you so much, Dr. Eva, for a nice presentation. And just about now, we are until the end of our session, and I'll just ask you to make a final call. But I would like to congratulate KEMRI for such a nice work, and PAPERI for bringing this up so that also others can learn and maybe replicate or collaborate what has been done to also help Africa into precision medicine. So with all of this, what would be your final call?
I would
Dr.
say
Eva.
the future of human genetics in and genomics in Africa is bright. We have what it takes. We have the human resource. It might not be enough, but we are somewhere. We need to work together. We need to move from our silos. We need to think less as countries, but more as a continent, and to have a common agenda. Yeah, let's work together. Where people are stronger, can we assist each other? If you have more capacity in clinical genetics, can we build our networks so that we can know where to get help and get this scaling or precision medicine to become a reality in our continent based on the data sets that we have generated. That would be my take, my final word. Yeah. Thank you.
Thank you so much, Dr. Eva. You are right. Thank you.
Yeah.
Now we should close. Hagai, can you hear me? Hagai? It's freezing now. Okay, let me close the session. Thank you again, Dr. Eva, for answering the question and for your excellent presentation. It was a pleasure to have you with us. Thank you all for attending. We hope you found it informative and enjoyable. Have a great day. Goodbye. Thank you. Bye-bye.
Thank you and goodbye. Thank you.